organizing committee for the invitation to this meeting and to be a speaker today. And I will be talking about leishmaniasis, and I will give an overview of the disease, and also we'll talk about the vaccine development, and finally, in the last couple of slides, I will mention the type of research that we are developing in here in Al Faisal. So leishmaniasis is a complex of infections caused by a unicellular flagellar parasite called leishmania. The disease has different clinical presentations, and here is shown 3 of them, the main of them. One is the visceral leishmaniasis caused by Leishmania donovani, mainly. And this parasite affects the liver and the spleen, and is one of the most dangerous disease in this disease group. The second group of that's shown here is the one causing cutaneous leishmaniasis. The disease localizes in the skin, different places on the skin, and causes this type of lesions. The third group here is the mucocutaneous leishmaniasis, which exists mainly in Central and South America, and the parasite localizes mainly in the soft tissue of the mouth and in the cartilage, and causes severe disfiguration of the person. The parasite is transmitted through flies. They are called sandflies. And in the Americas, the Lutzomia group is the one transmitting this parasite. And in the old world, Phlebotomus is the vector of the disease. These flies are very tiny, 2-3 millimeters in size, and they are hairy, and the bite is very painful. The parasite has 2 forms. The one form shown here on the left is called promastigotes, stadium of promastigotes. It is the stadium that lives inside the fly. And it has a long flagellum that allows the parasite to move inside the gut of the fly, and allows to move to the mouth parts of the vector. On the right side, we can see a macrophage with the nucleus and the cytoplasm is full of parasites. This is another stadium of the parasite which lacks flagellum, and is the one that we can find again inside tissues during the diagnosis. The transmission occurs when the parasite living inside the gut of the fly is transmitted to this, transmitted to the reservoirs in the wild, or to humans, which are accidental hosts. The parasite is taken up by macrophages, and the parasite divides inside the macrophages and causes the disease that I just mentioned. When the fly feeds on humans or reservoirs infected, the fly takes the parasite inside the mosquito, the fly, the a parasite transformed into this flagellated form and ready to infect new hosts. The distribution of the disease has a worldwide distribution. And it exists in Central and South America, in Asia, Middle East, Africa, but is most concentrated in the case of visceral leishmaniasis in 6 different countries. In Africa, in Sudan, South Sudan, Ethiopia, in Asia, in India, Bangladesh, and in the Americas, in Brazil. As you can see this in the dark colors, these countries contribute to around 90% of the transmission, as reported. In the case of cutaneous leishmaniasis, the distribution is also worldwide. But the main reported cases have been found in Afghanistan, in the dark areas here, Afghanistan, Pakistan, Iran, and Ethiopia, in Algeria, and in the Americas, in Brazil, Colombia, and Peru. The disease, it is said that the disease can affect a quarter of the population worldwide because it exists in areas, highly populated areas. Eventually, 12 million people are infected at one time point. There is no vaccine, and the drugs to treat the disease are not 100% effective. And as we can see later, there are other problems associated with these drugs. In the Middle East, there has been a surge on the amount of cases, especially in countries that have been going through a severe war, such as Syria, as shown here. In 2011, the Syrian number of cases rose to 60,000 cases that year. Other countries in the Middle East, the transmission is quite stable, but the number of cases remain high. In Saudi Arabia, fortunately, the disease has been going down, or the number of cases has been going down since the Ministry of Health took measures to reduce the disease cases from 18,000 in 1983 to approximately 1,500 in 2015. But 1,500 cases in Saudi Arabia is considered still a high number of cases. And in addition, it is believed that the disease cases are underreported. These are the regions of the Saudi Arabia where the disease is mostly concentrated. As you can see in dark yellow here, the disease exists mostly in the central of Saudi Arabia and in the east and in the east regions. Fortunately or unfortunately, the disease hasn't spread widely in the whole country. And also to point out that in the Makkah region, it has declared disease-free. The elements involved in the disease as shown here, essentially 2 parasites are responsible for the disease, Leishmania major and Leishmania tropica. Leishmania major is mostly transmitted by the vector Phlebotomus papatassi. And Leishmania tropica is mainly involved, Phlebotomus sergenti is mostly involved in transmission of Leishmania tropica. And there are 2 rodents involved in the transmission, in the reservoirs of this parasite. They are Psammomis obesus and the mirions. These are rodents living in caves and places in the desert, and they can live for many days, even weeks, without food or water. Interesting that Psammomis obesus is at present one of the animal models to study obesity in humans. And the reason is that this rodent, when it has the chance, it eats a lot and becomes obese. These are the cutaneous lesions induced or caused by the transmission in Saudi Arabia. There are multiple lesions formed in the arms, in the neck or face or legs. But there is also single lesions being reported. Current treatment. Treatment for leishmaniasis in general is based on very few or limited group of drugs. 4 of them are shown here, which are the most common. But sodium stibogluconate is the most commonly used. However, any of these drugs has a limited effect. They are, in some patients, they are useless, and many patients, and some other patients, the response is very poor. In addition, these drugs are toxic, usually take weeks for the treatment. And the most scary problem with these drugs is that the leishmania has developed resistance against most of these drugs. So obviously there is an urgent necessity of a vaccine that can prevent the transmission or the disease transmission. So various attempts have been taken in order to develop a vaccine. Initially, the vaccine was based on live attenuated parasites or killed parasites or parts of the parasite. This approach, although gave some positive results, become impossible to set up as a vaccine. And the reason is that the results are variable and the standardization of the vaccine is impossible, it's very difficult. So in the last few years, in the last 20 years, I would say, the scientists have been looking into the other approaches, such as using molecular biology technologies to develop vaccines. One of those approaches is based on chimeric vaccines, in which the leishmania antigens are placed in vectors, such as viral vectors, and use this vector as a vaccine. Other approaches shown here involved the use of various molecules, various proteins of the parasite to make a kind of polyvalent vaccine. But recently, in the last 10 years, there have been a lot of research on the recombinant vaccines. In this case, a vaccine called LaICH-F1 has been very advanced in the study of immunization using this vaccine. So the vaccine was developed by the Infectious Disease Research Institute in Seattle, and it contains 3 antigens. The antigens are conserved across different Leishmania species, and therefore promise to be used, can be used in different disease presentations. And the vaccine has been tested in humans in cutaneous leishmaniasis, visceral leishmaniasis, and mucocutaneous leishmaniasis in the Americans. The vaccine has been shown to be safe, tolerated by humans, and most importantly, develops a cell based on CD4 T cells producing Th1 cytokine, which is the one that can shown to be effective against to control leishmaniasis in animal models, and there is also correlation in the humans. There are other different antigens that have been recombinant antigens that have been isolated, identified, isolated, and studied, and many of these are protective antigens. So there is a hope that a new vaccine against leishmaniasis can be developed in the near future. However, there is a little problem. Recombinant leishmania vaccines need the presence of vaccine adjuvants. So these adjuvants can help, can trigger boosting the CD4 T cell immunity. As I mentioned, is the immunity required for the leishmania control. And in this search for adjuvants, there are many people, companies, and organizations looking for adjuvants that can boost the T cell immunity. And this brings us to the project that we are developing here in Saudi Arabia in our Department of Microbiology, Immunology, and is based on the following scheme. We are looking for adjuvants shown in the Sorry, this is stupid. How you can go back to the last one? So our project is trying to identify the adjuvants that can trigger T cell immunity. Thank you. In the left panel here, it shows what it says TLR-mediated adjuvants. We are using modern concepts in immunology to identify adjuvants. And the concept of dendritic cell being triggered by adjuvants has become very important. So in this case, dendritic cells treated with adjuvants can present antigen, effectively present antigen to T cells. And the T cells, depending on the cytokines produced by the dendritic cells, the T cells can become either CD4 protective against leishmaniasis, or can be regulatory T cells, etc. So we have the model of using toll receptor mediated adjuvants as tests for this new method. Once we identify the adjuvants by using dendritic cells, then we are going to use these adjuvants with different antigens and test those antigens in a vaccine in mice or in other animal models. So my conclusion is that leishmaniasis remains a serious health problem worldwide, and particularly among poorest population. Leishmaniasis has a problem. there is no treatment and no vaccine available for any type of the leishmaniasis. However, significant amount of research shown that a prophylactic vaccine is possible, and but requires the efforts of many stakeholders and further research. Thank you.